everyone, my name is Silvia, and today I'm going to introduce a ligand receptor family, the neurotrophins and TRK receptors, in the context of neurodevelopment. Development of the nervous system undergoes a great expansion through neurogenesis and growth. It is known that in early development, many neurons are born but undergo apoptosis soon after. Then what promotes neuronal survival? What are the underlying molecules behind this process? In 1950s, Stanley Cohen's team made a serendipitous discovery with snake venom. When they added it to the dorsal root ganglion neurons from chick embryo, the neurons survived and grew projections. This later led to the discovery and purification of a neurotrophic factor, nerve growth factor, NGF. NGF was the first discovered member of the neurotrophin family, a class of proteins that play an essential role in the survival, development, and function of neurons. Other neurotrophin family members include the brain-derived neurotrophic factor, neurotrophin 3, and neurotrophin 4. NGF is critical for survival and maintenance of sympathetic and sensory neurons. After being released from the target cells, NGF binds and activates its receptor, TRKA, on the neuron. Brain-derived neurotrophic factor, BDNF, is a neurotrophic factor found originally in the brain, but is also expressed in the retina and motor neurons. BDNF plays a significant role in supporting the survival of existing neurons, and it also contributes to the growth and differentiation of new neurons and synapses through axonal and dendritic sprouting. It binds to the receptor TRKB. NT3 has similar functions as other neurotrophins in controlling growth and differentiation of sympathetic and sensory neurons. NT3 is unique in that it is able to activate two receptors, TRKC and TRKB. Lastly, NT4, also known as NT5, promotes growth of the sensory neurons in the periphery. It mainly binds to TRKB to initiate its signaling pathway. Now let's take a closer look at the TRK receptor family. Each neurotrophin has its specificity for a particular receptor. NGF binds preferentially to TRKA, BDNF and NT4 to TRKB, both of them actually with a lower affinity. Finally, NT3 is able to activate all of the TRK receptors, but it mainly binds to TRKC with a higher affinity. Here's a simplified structure of a TRK receptor. It has the leucine-rich motifs and IgG-like domains that regulate the strength and specificity of the binding between neurotrophins and the TRK receptors. Intracellularly, it also has a kinase domain that enables the activation of a signaling cascade for growth and survival. Now we have answers to these two questions. First, what are neurotrophins and TRK receptors, and what do they do? Neurotrophins are survival factors, and they interact with TRK receptors to promote the survival and growth of neurons. Second, where are they expressed? NGF, NT3, and NT4 are expressed in sympathetic and sensory neurons, while BDNF is primarily expressed in the brain. In the next half of the video, we will explore first how the signaling pathway mediates the survival and proliferation of the neurons during development, and second, when and what stage of development requires the action of these neurotrophins and TRK receptors by learning their specific roles in differentiation and functioning. The common pathway for neurotrophin and TRK receptor signaling is as follows. The binding of neurotrophin as dimers induces TRK receptor dimerization, resulting in trans-autophosphorylation of the TRK receptor. The activated TRK receptors then recruit and phosphorylate PLC gamma and SHC downstream. The interaction between TRK receptor and SHC in turn enhances the activity of PI3K and AKT. The phosphorylated SHC also activates RAS via GRB2 and SOS protein, which further promotes the ERK pathway. Here I have color-coded to show the different effects for the three main signaling pathways. They all lead to various transcriptional events, such as the induction of transcription factor CRAB, which promotes differentiation. PLC gamma signaling induces synaptic plasticity, and PI3K signaling promotes growth and survival. Now we have learned the molecular pathways of neurotrophin and TRK receptor signaling. Then when in development do these interactions play an essential role? Here is a developmental timeline to help you see the big picture. Early in development, these neurotrophins are involved in promoting survival and proliferation of the precursor neural cells. So for example, NGF promotes the proliferation of nociceptor neurons. BDNF and TRKB interactions affect the survival of CNS neurons, particularly the cholinergic neurons in basal forebrain. And lastly, NT3 and TRKC interactions promote the proliferation of neural crest cells. They continue to play a key role in regulating target innervation. For example, NGF controls the advance of sympathetic neuron growth cones, which was shown in experiments where these neurons could not reach their target without NGF, even when given enough trophic support. They are also involved in sensory neuron function and ocular dominance column formation. Both NT3 and BDNF are important in development of synapses between afferent and motor neurons, while BDNF and potentially NT4 also play a role in promoting ocular dominance column formation. Lastly, neurotrophin and TRK receptor interactions are relevant in synaptic plasticity, as it was shown in studies that loss of BDNF or TRKB reduced LTP significantly. In this video, we have covered the types of neurotrophins and TRK receptors, their expression in different parts of the nervous system, the signaling pathway, and their roles in development. Neurotrophins play a crucial role in promoting neuronal survival and growth, and currently researchers are trying to understand how neurotrophins protect neurons under pathological conditions for future translational research and drug development.